Les agradecemos por estar acá, a las personas que están presentes y a las personas que están conectadas por Zoom. Tengo la alegría de presentarles al doctor Gilles Cellier, ¿ya? Les voy a hacer una breve introducción acerca de su currículum, ¿ya? Él es ingeniero agrónomo, tiene un máster en desarrollo y mejoramiento de producción vegetal y es doctor en biología molecular. Actualmente él trabaja en la Agencia Nacional de Seguridad Alimentaria ANSES de Francia en el Laboratorio de Sanidad Vegetal, ¿ya? Y su proyecto actual es acerca de la diversidad y la epidemiología de Ralstonia solanacearum, el complejo de especies. Además, el doctor Cellier es editor asociado de la revista Fitopatología y pertenece a diversas redes de investigación para el desarrollo de protocolos tanto del complejo Ralstonia solanacearum como también de Wenlong. Y bueno, asociado a los diversos proyectos que él participa, tiene varias publicaciones relacionadas. Y hoy día nos va a presentar entonces su investigación, que es acerca de la epidemiología del complejo de especies de Ralstonia solanacearum.
Sí. Hola a todos, hola a todas. Estoy muy feliz de poder compartir a este seminario. And that'd be around that much of Spanish I know, so I'm sorry. The other sentence I know is El gato está sobre la mesa, but it's not going to help this presentation. So my name is Gilles Cellier. I'm working for the ANSES. Yeah. Okay, gracias. And the talk of today will be about Ralstonia solanacearum, the species complex, but most of us will understand more the underlying experiments behind and the technology used to assess its epidemiology and the population structure of this phytopathogen. Well, first, my agency is located in mainland France, but we are working not in this territory. I'm working in Réunion Island, just right here. So it's very far from France and even more farther than Chile. And we have to get busy on expertise and different field of phytopathology with all the different departments of France, which call our département ultramal, departamentos de France. So Réunion Island, why Réunion Island? Because I've done my PhD there. This is the laboratory I'm working in. We are hosted by CIRAD, another research center. And for us, ANSES, we are a reference and research laboratory with both analysis and also basic and applied science and fundamental researches. About Ralstonia, we have the chance to have on this island a hot and a cold tropical climates. And out of 4 species of this species complex, 3 are present in this island. That make us available to work in like P2 laboratory and not P3 for every strains we are working with. This is a great advantage. So the topo of today will introduce some definitions around molecular biology. We'll see some phylogenetics about the RSCC, Ralstonia solanacearum species complex, easier to say. We'll see 2 practical cases, one of a phylogeny based on a single gene, and one is an epidemiological study about the RSCC using different type of markers. We'll see that. And then we'll ask what can genomics bring to this phylogeny for this species complex. So first, we have to do that, I'm sorry. We have to read molecular epidemiology. It's a discipline that's using, of course, molecular stuff or genetic markers to trace the development of a disease within a population. The definition of population can be different from one field to another, okay? For us, it's like an assembly of strain, around 15, 20 strains or clones that represent similar traits in the environment. So to understand the transmission, as well as the population structure, evolution of an organism. The phylogenetic analysis is very different from this approach, actually, but can be complementary. It allows to use marker to determine the genetic relatedness of strains from different source, geographic location, and or even different period time, inferring evolutionary relationships. For this means, I use this representation from Romagnac and Al, which talk about classification, okay, and this kind of typing, actually. So classification, it's like a top-down. So we start with the species, and when we go down, it's straight classification using species subdivisions into like clonal groups, and each potentially representing descendant. This is a relationship among ancestors. The one is known. Typing, it's what we actually do most of the time. We find a strain in the environment, and we're trying to find out what is it. Is it related to this clone, this clone, this clone, or something different? So this is called a bottom-up strategy that can assign a related variant to clones that we call further strains, which represents the cloning structure. It can only rarely elucidate the phylogenetic relationship between different clonal grouping. And we don't use the same markers for going through classification or through typing. So one appropriate goal for molecular biology might be to define stable groups among all this diversity and all the things we can do, and we can only assess through our markers. We're blind to this. So this group of organisms with epidemiological patterns, this is really important because the biology has to fit with the molecular biology. So classification and typing are necessary also for risk assessments and surveillance. And of course, when we work on a given pathosystem, we need to assess a proper question that has to be linked with an appropriate scale of the classification, the typing for this studied organism. So we use genetic markers, of course. Genetic markers are heritable character, multiple stage that can be drawn at each character. We can use a single marker, just like a gene we're going to talk about, or a pool of selected markers. And we're going to talk about this also later in this presentation. So we use like a trade-off between the accuracy and the practicality of the methods, because we cannot use like hundreds and hundreds and hundreds of markers and spend like 3 years on only one strain. It's impossible. So those techniques can be multi-locus based, and this is all techniques, just like RAPD, AFLP, or single locus DNA based. This is the nowadays technique you guys are certainly using, like microsatellites and single copy nuclear DNA reagents, etc. Each markers have their own evolution time. We call that clock rate, okay? Different clock rates are necessary for a given research. If we use only one gene, we have to assess this clock rate in order to see if we are working at the species level or the clone level. And in between, have a lot of shades of gray. And when we use a pool of different markers, we can use different clock rates according to the different markers and to bind their power and to make sure that we will resolve the question asked. We have also the high mutation rates, which are appropriate for recently evolved species within the diversity. So this is a representation of mutation rate according to population genetic diversity and marker utility. So if we move around this 3D graph, we can see that some markers are good for resolving deep roots among the whole species, and some of them are really good to resolve terminal clusters. So a mix of them, or using only one category out of another, will tell us different story of the system we're working with. A correct sensitivity also has to be linked with the scientific question. This is trivial, actually. So we have to know what we're working with. See? Okay. And the analytical signal can be also jammed when there is too little or too much information. So we can produce a large amount of data, and we don't know what to do with that because the noise, the background noise will be too important for what we're going to talk to. Or when entities are really different, when we try to look two different species and we think it's the same species when we have a bunch of different answers we cannot even understand. So Ralstonia, what is it? This is a phytopathogenic bacteria. This is an example of it, plate on a petri dish. It is the second most important phytopathogenic bacteria across the world, according to some publications. And it considers, of course, as quarantine pest in many, many different countries. Rhizospheric means that it lives close to the roots, but it can live also in water and different type of environment. It can be hosted by 54 different botanical family, from bananas to eucalypts to potatoes and tomatoes. This is astonishing. It has a worldwide distribution. It's present in Chile with a host range and high economical and social impact. We have 3 different species we're going to talk about: Ralstonia solanacearum, pseudo-solanacearum, and CZG. I was referring in the first slide to phylotypes. This is we're going to see that in the next slide. This is the phylogenetic groups that can be seen on this diversity. 4 phylotypes, and the subdivision is called Sécurité, around 70 of them. So throughout the time, we use different techniques to appropriate this diversity and this complexity. And we start, of course, with race and biovar that was based on hosts. So if, for example, this bacteria was wilting mulberry, that would be one race. If this strain can host in banana, that will be another race. And we thought that it was okay at this time, but then the genomic era with the PCR came, and we ordered that with the phylotype and the Sécurité since 2005 around. And then came a lot of different advances, like the first genome in 2002, the first pangenome, genomic microarray, the MLSA scheme, MLVA scheme. It goes very fast. And thanks to what? To the genomic, of course. The genomes available increased a lot throughout the years and reached a high level. This is one of the most phytopathogenic bacteria that is sequenced today. So phylogenetics, I'm talking a lot about this kind of things. So we're going to see the species complex. The 3 species are here, then the 4 phylotypes. One species is gathering 2 other phylotypes. Here is the different techniques that we can use to assess this. So techniques means markers. Here we use some PCR, like some phylogeny can be used on single gene or multiple gene, just like in MLSA, MLST approach. And we can go down to Sécurité and then the ST, MT, MLAG. I will explain that a little bit later. But you have to understand that this is a hierarchy among the classification, okay, from the top to the bottom. And what we found in nature is usually this. And then we goes up and we seek for different markers to assess what is it exactly in the field we found. So this is well, it's not used for this presentation, but it's okay for this presentation. So why are we sure that this phylogeny is the good one? Because we only see what the molecular biology tells us. We amplify a gene, we sequence it, or we have a restriction through enzymes making different bands or a different size of amplification. And we can use like one gene, EGL, or the ETS, and we see this classification in 4 phylotypes. That is good. We can use another gene, the HRPB, and we see also this classification in 4 phylotypes, etc., with MUT-S and many other genes. Those are housekeeping genes, meaning that the pressure of evolution is low compared to other genes, and we can assess the state of the species. And of course, if you use genomes, for instance, we see the same thing. So we are pretty sure that the diversity represented here is kind of the true representation of the natural state of this complex species. So different approach using different technology leads to the same point. we want to seek for the diversity order, natural order, we approach with different markers and we have the results. If every results are the same, bingo, we're good. So the reconstruction of the relationship through a phylogenetic inference is kind of difficult. Just like I told you many times, we have to approach, and these approximation methods are using different techniques, either the markers, the techniques itself, the statistics, or the algorithm we use, and it can be biased. So we have to make sure that all these techniques is used properly throughout your experiment. So inferring a phylogenetic tree is about actually reconstructing, English is difficult, the evolutionary relationships between the taxonomic unit and through an estimation. It's always an estimation. You won't see the true, okay? So this required a proper alignment of the sequence, and then we can use different classification methods, so algorithm, we can call them whatever, and we have to orient the tree to roots, etc., and to assess the nodes' strengths among everything. So we are going to see a practical case on the easy one, the EGL endoglucanase, which is the gene involved in the pathogenicity in this species complex. And that is weird to use pathogenic related genes to use this because it's usually evolving quite fast to interfere with the host resistance, the host response, etc. But it works pretty well. So what we need for a proper phylogeny inference through the OGL or whatever genes you're using, it's sequence, proper alignment, evolution model, and some algorithm that are going to build and infer these trees you're going to, you're familiar with, I hope. So we can use different techniques. Here is just an example, crustal or muscle, etc. Which is really important is the model of evolution, because how to best fit a model of evolution with all the rates that change, is a G changed by a T has the same value as a A for a G, etc. We don't know. Those models will assess this according to our sequences and properly. So a lot of models exist. So you see publications, there are like 20, 30, 50 different models. So both are existing for amino acids or nucleotide sequences. Simple model are best suited for closely related taxonomic units. So two clones, simple model, you will assess if they're different or it is the same. For more complex model and adapt for stronger evolutionary distances, of course, you will have to use additional parameters to this. So to complexify everything, and it can be sometimes tricky, okay? Because you introduces biases. And biases over time will differentiate wrongly two strains, but are actually the same cluster. So you can say, okay, I'm using the more complex model, so I'm sure that it's fitting perfectly and I'm good with it. But these biases won't help you actually to accurately describing the evolutionary topology. So what I propose is to use this kind of software, which will assess, according to an alignment of the sequence, which model is best fitted for you. It's really simple to use, and you won't mistake your research and your data. And at the end, using an ML algorithm, for instance, will help you to reconstruct this phylogeny. It uses the likelihood value. So how likely this separation between two strains, two clone or whatever, is true or not. So it will assess and find a tree that is maximizing this probability of observing the data for a particular model of evolution. This is truly powerful, and this is really working very great. And we can then, using different type of strains, assess, of course, the phylogenetic structure we know with 3 species, okay? And using a correct assessment also of the node through a different test, SRT or LET or whatever, you have to assess if the node is here and how robust it will be, okay? And for the routing, well, this is particular to Ralstonia, because the 3 species, usually you don't work with 3 species together in a single tree. You work with one species and you see and you assess the diversity in one species, one another, and another again. This is a typical representation we like to see in publication, but to make it right, you should only focus on one species and use the other related species as outgroup to orient the tree and to assess the length of the tips of different strains. Come in. It's the break. Get that water. So now we're going to see the use of some marker for the molecular epidemiology. So those are two terms that we use frequently, but we don't know how to relate to them because, well, thanks to COVID, we have a lot of different knowledge of how epidemiology works. But for human and for plant pathogens, this is not quite the same. So as we've seen with the EGL, we can have a different clustering called Sécurité, okay? One gene, we have this kind of clustering, different Sécurité within the phenotype number 1. Phenotype 1, 16 is the Sécurité. What if we use not one gene, but like 7 or different genes that can be useful for assessing the species diversity? So we can use, for instance, 7 different genes, including the EGL sequence. And we can see that some Sécurité are splitting into what we call ST, sequence type. This is a different approach. So the marker are more refined to assess the diversity, but it's like not enough because the MLST, MLSA techniques are designed to see the species actually delineation and inside it, but not going further. What if we use a different approach and use this MLST marker? And then, amazing. So we have like the MT, multilocus type, using 13 marker. So it's not because we're using a lot of markers, we'll see a big diversity. It depends on the nature of the marker. Here, the clock rates is much higher than here and here. So, but using only EGL is good. MLST, you're working like 7 times more and you have to refine it like a little bit. Here, 13 different markers and you have, look at this, whole bunch of diversity and everything. This is more powerful. We call that the discriminatory power, okay? And so using proper markers, proper approach, without working that much, you can achieve this kind of diversity discovering. And look how logic it is from the EGL to this. There is no splitting of this, for instance, into this or into this. This is completely logic and hierarchically organized. I like it that much. So MT, multilocus type, combination of different alleles from different loci located at the same genome transmitted together because it is scattered around the genome. Here is the strain GMI 1000, well-known strains. And Ralstonia have 2 different replicants. One is called the chromosome, not the other the megaplasmid. And we designed what on these genomes, what we call VNTR. Variable number or tandem repeat. Tandem repeat, what is it? It is those kind of small sequence like TT, CCC, GG, repeated n times. It's like the genome is stuttering, many different repetition of this, and the number of repetition can be assessed through like primers, okay, pretty simple, fluorochrome, and then we see the size. And according to the size of the offsets that you design everything, the size of the primers, blah, 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 you can assess pretty easily the number of repetition that you have from strain A, for instance, strain B, on different VNTR. So tandem repeat 1, 2, 3, 4, and you have a code, like a barcode. It's not barcoding, but then you have like 7, 10, 15, 25 different markers, and you have this cool code. And with this, you can work very deep into resolving the diversity of the bacteria. But before working on these markers, which are awesome, you need to properly done your survey in the field. You cannot go and take one here, one there, and say, okay, I'm done. I'm good with the markers. And as he said, I will be published in Nature. It's not true. So We go in the field and we look for populations. So we need to sample a lot of different plants and hope they're harboring the bacteria you're looking for. It's working with various assay. And then with the GPS, we take the stem and etc. We isolate the colony, one colony for one plant out of 30 in the fields, and we have then different approach. Then we first type, then we assign a phylogenetic stuff, and we work on population genomics. But this amount of strains and the capacity for you to gather in the lab population, a gathering of multiple strains from a single location is really important. So we're going to talk about this scheme called MLVA using 13 markers on the phenotype 1. And I assessed this scheme on a huge collection of strains. I know it's surprising, me too. So what we do, we went to the field and collect different samples all across like Africa and Réunion Island, Mauritius, Seychelles, Comoros, Madagascar, well, whatever. It's all the Africa and Indian Ocean. And we sample this amount of strains. It is crazy. We select the 13 markers, and this is for the geeks. We don't need that right now. What we are going to assess here is not to say that, okay, our scheme is the best. It is perfect. We have to publish it and that's it. But to assess the markers complexity and the ability to resolve different, how do you say that, refining into the diversity of the pathosystem. Because one marker won't be the same as the other marker, etc. And of course, everybody says that in their publication that their system is the best, but we have to assess it. And we see that all the loci displayed actually polyformism, which is kind of cool to see the diversity, but are not equally informative. And that we are going to assess it and to see it later. First of all, the 13 markers allow us to accomplish what we are going to. It's like, using all this, we have quite the near saturation on the diversity, meaning that using this 13 marker, when we assess the diversity, we are quite sure, but quite sure we have 100% diversity we're going to see in the prospect, in the survey. The first result of this huge survey on 2,670 isolates selected is that like there is one Sécurité that is hugely predominant. About half of the strains belong to this Sécurité, 31. Perfect. Remember the Sécurité different classification, then we can go further with the MLVA scheme. Then the Sécurité 18, 33. Well, the most important one, I spot them with the star. And here is a representation of the different marker used, the VNTR, in this direction, and the number of repetition by frequency. So repetition of the different tandem repeats. And of course, the higher the number is, the better it is to assess the diversity. That's what we call the polymorphism. But we see that most of the marker have a low mean of repetition per locus. It was assessed like below 5, you won't see that much things. It's not a good news for this. And also the polymorphism among some markers appeared lineage specific. Here it's Sécurité specific. So for instance, that we saw that Sécurité 31, which is the largest amount of strains we gathered, most of the markers have low repetition. So the power of this discriminatory among this specific lineage is kind of low. And I was talking about polymorphism about some markers, lineage specific. That means that this marker, for instance, is highly polymorphism, but only for this one. This marker only for this Sécurité. This marker only for this Sécurité. So, but the good news is that for the Sécurité 18, which is found mainly in Madagascar, for instance, and in Europe, we have a lot of diversity. So the message here is that know your tool, know how to assess your tool to understand how confident you will be to address your conclusions in a scientific paper. Because if we haven't done this, we would have said that just because of these results, okay, we are seeing all the diversity what we need. It's perfect. Let's go for it. But seeing this, we have to draw conclusion with a subtle approach, okay, on different strains. Then we use a DAPC approach, which is statistical, to cluster the different strains and assess if the markers are explaining or not the diversity. So on this axis, you have 73% of the diversity represented and explained, and this one, 30%. So more than 90% of the diversity here. Good job. And we have 4 clusters. 1 clusters, 2 clusters, 3 clusters, and the garbage cluster here, gathering a lot of different strains of Sécurité, different Sécurité. And my point here is to go deep into the data and to look, is it good or not? Because this one is the cluster mainly represented by Sécurité 31. Okay, but 31 is also among this right here. So why does Sécurité 31 here are in the cluster 4 and the rest are in cluster 2? Because you have to understand that those suspicious isolates are like misplaced by DAPC or by the Sécurité system. And we found out, to be quick, that there is in the gene called EGL, what we used for Sécurité estimation, there are some recombinations that will hamper the correct assignment of the strains. And in the email via, we have indels, insertion deletions in the sequence. That means that we saw like 5 repetitions, but actually that's an indel. There is 2 repetitions more because the evolution process. That's it. But we have to spot this, the weakness, the strength, and the weakness of every system. But this is very beautiful. We have like a huge grouping of the strains together. This is a representation of clusters. This is not phylogenetic because on one point you can have more than 3 link. Okay, this is like a cycling representation of the diversity. And if we map the color of the DAPC, we can see that it totally respects what we see. Sécurité 18, Sécurité 31, the 33 here, so the different DAPC colors, and the garbage group, which is here. And the huge diversity here among different countries is gathered into only one cluster. 50% of strains are here. And this is well separated. So we have a good job on these markers and we can draw conclusions. Then we run some intelligent stuff about epidemiology assessment using statistical approach with your G'-ST and RST score. And we can draw within populations the link if they are different or not. If there is a link, that means a line here, it means the population are not different. And we can see that remote population from Côte d'Ivoire and Madagascar, not Madagascar, sorry, Réunion and Mauritius, they're the same strain. How to explain that that much far, they're the same? But we have to keep in mind that this for the APC 33, 1 and 2, which is 33 and 31, the markers were not really good into going deep into differentiating strains. So it can be artifactual. But if we see the genomes, they're actually lack of genetic diversity, though they're kind of the same. So the question now is to assess how do those 2 different groups so successful in a different remote place, and how did they replace the strains they were before and contaminated the countries? So greater dispersion through plant material or greater adaptive fitness. We're going to seek that. So here are the conclusions or the different stuff we saw in this example of epidemiology microbiology. So we assess the system here, that it's often related to Sécurité than the true representation of the nature, except for Sécurité 18. Limitation in the Sécurité typing system, so problem with assignation. We have to check that also. Phenotype 131, major causal strain of the bacterial within the Africa and Southwest Indian Ocean regions. And the strains you have here in Chile are 2B1. They're born here in the Andes. This is a big issue in Europe because it can adapt to a cold temperature. But this 131 situation was never foreseen before and firstly mentioned only recently, like 10 years ago. And it represents a low genetic diversity that could hamper epidemiological analysis. Our system at first, the strain we're working on are not very diverse. So using a lot of markers will be necessary actually, and with high clock rates to differentiate them accordingly. Last few slides, I promise it's almost done. It's the use of the genomics, because we're going, we've seen that previously, that it was useful, but useful for what? Because it says in the project, but no, not only. It can complement, actually. For me, it will not replace, but complements the older approach that we used already. For instance, species delineation. We have now 3 species in the Ralstonia solanacearum species complex. Before we used the DDH, which is hybridation of DNA. It was fastidious as hell, and only few labs can do it. And so species complex was defined later because we foreseen that there is a problem within the species within this species complex. 2 diverse strains cannot be one species at once. Recently, Nationale used this approach, phenotypic characterization, whole-set fatty acid, and DDH, which is the old method. They trimmed a lot, they worked a lot, and they end up by publishing first, congrats, on 3 different species, but that was the old way. And Prior and al., like a year later, they used the whole genome sequencing, proteomic analysis, and metabolic characterization, and they end up with the same conclusion. So why really using these methods that are taking a lot of time, why we can use the genome for the same answer? This is the purpose and the message of this slide. So yes, you've seen this image with the 2 phenotype here, the 2 with the 3 species. And yes, we can use the genome. It has a high correlation with the DDH standard because it has to move somehow from one technique to another to see and to make the science go further. So this is a faster approach, less expensive and less prone to technical and human error also. So yes, they are really available for species delineation, and that's a great advancement. I don't know if you realize, because bacteria are been working with scientists for so many years, and we'll still have to get them out of the plant, plate them, and have this physical bacteria not in your hand, because it's not good, but in the plates. And some bacteria are not able to be cultivated into plates. So how to deal with that? So this ancient kind of thinking is going to evolve, and I hope it's going faster and proper to have this kind of representation. And of course, to define the threshold, because we use an ANI, average nucleotide identity here, algorithm. So one algorithm makes some results. We have to assess the algorithm. And of course, to delineate species, we have the threshold that is still going into a discussion. So genomics open more aspects in population study, because we have alignment, we have phylogenetics. Now we are expert in this, I'm pretty sure. And now we end up with population genetics. We go further than the phylogenetics, and we can make everything like this. Population demography history, identification modes, direction, and chain of transmission, tracking spatial and temporal distribution. For instance, you're finding a strain at a moment, and 5 years, 10 years later, you're going back to this field, you retrieve this strain, has it evolved or not? Here is the answer. And you can characterize the past epidemiological dynamics and estimate different parameters like this mechanism, important HET, genes, etc. Population history, has it grown, bottleneck or not, and different stuff. So these approaches are highly complementary. Okay, this is the key. So we have to use a proper methodology, will help answering the question, scientific question, okay? And every inferring methodology is an approximation, okay? It has weaknesses, but it has also advantages. We need to push forward wider adoption of genomics, of course, and to use clean data, shared also. Don't keep your data for yourself, please. Publish it in international basis and make it used by other lab, collaborate, and everything will be better for the science. And we need to use also multiple methods that examine actually different scale of the problem. Okay, this is really important to describe a complex given pathosystem, okay, acting in a complementary fashion. I promise it's almost done. Thank you. to all the collaborators actually who helped to this project, and two special thanks also to Doctora Miriam Valenzuela to host me here in Chile, and to Ingrid Nicole Vasconez Hurtado, who is going to be a doctor, I hope, next week. Wish her luck. And thank you for your kind attention. Voilà.
Ahora si alguien quiere hacer una pregunta en español o en inglés.
No español. En francés, por favor. So do not hesitate if you haven't understand something, or if you want to go further, or if you want to keep quiet, it's possible.
Las personas que están conectadas en Zoom, si las quieren hacer por el chat.
So keep in mind that all the techniques presented here is not only for Ralstonia, it can be used for different bacteria, viruses, and etc. This is the approach. You have to develop something, make sure that this something that you see is actually the natural things that happen or occur in nature, in environment, and then you can draw conclusions, okay? And respect deontologically the science that you're giving to others on papers. Voilà.
Eduardo?
Aside from average nucleotide identity,
Mhm.
have you ever tried to use any other whole genome related with metric?
Yeah.
I understand there's also like digital EDH, and do you have similar results?
Exactly.
Yeah.
But ANI is very trendy right now, and a lot of people are working on it. Of course, there are large amount of different algorithms, but it's like company. Everything is growing its company, and then it's getting down, and one last. The ANI has the advantage that it's a fast algorithm. It can be applied on multiple genomes, huge genomes. Imagine the genomes of the corn. Really, it is impressive. This is only 5 megabytes. It's not that much. But the advantage of the ANI also is like it was developed by Constantidis, and now we can use for species delineation. And we found recently another threshold for intra-species delineation, the 99.5%. And that makes things way easier than using different approach. Well, the MLVA approach is kind of also hard to achieve, and using genomes and good bioinformaticians, it can be great. But it's up to you to use the algorithm you want, as soon as it makes sense on the biological side. Recently, we have one of the phylotype that is renamed to another species using the wrong thresholds, because it was cool, because they have pressure to publish, and they have made a sensational paper. Great. But then all the scientific community come after them and says, no, come on, look at your results. This threshold is not good. If we calculate with these thresholds, this is not a new species. It's just this. So the sensational things can be achieved with different algorithms, but true papers are different. Thank you.
Tenemos una pregunta del chat.
Sí.
De José Ramírez, que dice, have you seen which phenotype are supervised most virulent in pepper?
It depends on the species on pepper, sorry. Because the plants, it's a host bacteria interaction. So on those two components, you have also the environment. Environmental components were like temperature, hygrometry, stress of the plants, etc., UV, whatever it is. So to answer the question, I don't know, but there are a lot. There are a lot, sadly. So yeah. We can find different strain. The phenotype 3 is only restricted in Africa. So at least you're safe here in Chile. We can find here the Ralstonia solanacearum, the 2B1. Yeah, why in France right now, the problem is with the phenotype 1, solanacearum. And yes, they all wilt the pepper. It's a solanaceous plants. And the 4 phenotypes can wilt the solanaceous plants.
But pepper is more sensitive to compare with tomato and potato, or less sensitive?
The advantage of pepper is that it has in their tissue a lot of lignin, which makes the plants very hard and resists the loss of water going through the vessels. Ralstonia is infecting through roots, by wounds, etc., and blocking the water efflux. So the water does not go up. So potato, tomato, less lignin, they will just wilt. This is the wilting disease. But for like pepper, they can be straight, they can bear fruits without that much damage. But there will be, of course, disease in the fields. But to compare the two, like potatoes and pepper, you will have less loss in pepper than in because of the plant structure. That's only this. Only this.
Eduardo Galvez, do you have information of the transmission of Ralstonia species complex? The transmission could be by seed, tomato or pepper seeds?
No, it's not. No, it's not. It's a long time that we're trying to assess the transmission through seeds. There is no proof yet, but we're like in science. Does not mean because you haven't proved the non-transmissibility by seed that it does not exist. But for now, we haven't proved it through different and many approaches. So transmission of Ralstonia is not in the air, it's in the ground. It's through material that cuts, for instance, banana with the ooze. If you remember on the plates, this is really oozing, disgusting. And you have this also, so grounds, material for cuttings. You have the plants, because some plants can harbor, just like the pepper, Ralstonia without showing any symptoms. This is latent infection, not latent infection. And this latent infection, so it's like for us during the COVID that some people have the disease, but they're not showing any symptoms, and then they can contaminate a bunch of people. So this is the same case also for Ralstonia. That's why it was introduced from, for instance, Africa to Europe. That's why 2B1 is distributing so widely across the world, because this is not a true seed, this is a potato, okay? They can harbor the bacteria without showing any symptoms. Then you sell it and it's prey. It remain in the soil, and then it's continuing. But the water also is a big issue for Ralstonia, because it can resist without any source of nutrients for dozens of years. It is impressive, even in the soil. For me, for instance, in the soil, we have a lot of sugarcane in Réunion Island, 15 years of sugarcane. They cut down sugarcane because it has to renew the stock, of course. And they planted, meanwhile, tomatoes. 15 years of sugarcane. This is a non-host for Ralstonia. Tomatoes grow, 70% of the plants died. No host, and it has survived, and it's pretty good.
Vanessa Ayala, ¿quiere hacer una pregunta? Parece.
Hola, ¿me escuchas?
Sí.
Sí.
Hi, thanks for your presentation. It was really interesting. I
Thank you.
have a couple of questions. I'll keep it short. What characteristic do you think of the species complex do you consider most important to take in account for our future, considering that we'll have to face climate change? And in the same line, what recommendations could you give to our Chilean researchers regarding the dispersion of these bacteria in our territory and the high susceptibility that important Chilean crops such as tomato and potatoes could have?
Of course, every country is exchanging goods and materials and plants legally or illegally. For instance, when you go on a flight and you have like honey, or you have like different plants, and you say, "Oh, cool! I'm going to offer like seeds to my friends," and you transmit this disease with the seeds or with the plants you carry or whatever. Actually, so the exchange, the global exchange, will enhance the communication points between different countries. Right now, we only found this, for instance, species of Ralstonia in Chile. But of course, global exchange and the global warming also will bring more strains, that is for sure, and tropical strains here in Chile or elsewhere in the world. And the main thing is like just wash your hands, but do the same with the farmers, that the seeds, like potato seeds, they're true seeds, not true seeds, but like potato tubers or different plants they have must be cleaned. So it's like a seal of approval that those plants are actually free of several pathogens. That's why we're working with the Ministry of Agriculture in France and different other Ministry of Agriculture to make sure that a given plants, I don't know how to say low, like that you import a bunch of plants, they are free of certain disease. And the diagnostic is really important to this. So fund the researchers, help the farmers with good practice also. And please do not do 40 years of tomato in the same field. It won't work. Okay, as soon as possible, a pathogen is entering to your field, you will make the population grow, of course, because you feed it. And this is called cultural rotation among field. You plant a horse culture, maybe it will give you money, that's good. But if you continue like this, your yield will decrease and you will increase the population of the bad bacteria or virus. So use a non-host, different type of plants if you can, and it will shut down the cycle of the pathogen. It works the same with virus, for the same with insects, it was the same with many different things. But like monocultural thing, it's an aberration for the environment and for the job of a farmer. This is not the way to do.
Thank you.
You're welcome.
Another question from Jose Ramirez. It is possible that a strain can lose its megaplasmid and become an alligator strain?
Losing a megaplasmid is like losing a vital organ for us. There are two mandatory replicants in this bacteria, and it's not possible. But there are plasmids, accessory plasmid in their genomes that can confer resistance to some antibiotics or advantage into the fitness. And this can be gain or loss among the species. But no, the two replicants are mandatory for the survival of the bacteria. It has essential genes on the two replicants. And this brings this observation that if essential genes are found into those two replicants, that means the separation of the genomes into two appeared like very long time ago in an ancestral state of the Ralstonia. And we can find this in the whole species, the 4 different species, 3. So that means 4 phylotype, 3 different species. So that means that the state before diversification of the Ralstonia into a species complex, the replicants were already separated. This is very ancestral character.
¿Alguna pregunta del público? Related with this question, how is Ralstonia could be this its virulence?
Yeah, it happens. It happens when you culture it for a long time, for instance, on a plate. When the temperatures drop too much, when there is a stress, particular environmental stress, it will lose its virulence. That is for sure.
How would that?
I don't know. I don't know. Maybe some other bacteria made this specific phytopathogen lose its virulence. If they are producing, for instance, volatile organic compounds, I don't know, for instance. This is a huge war in the soil. This is a huge war in the plants. Can you imagine? This is a xylem-limited bacteria. There is nothing in the xylem but water and some minerals. And the bacteria is growing at a population which can reach like 10 to the 9th CFU per milligram of tissue. This is astonishing. So how to fight against this? I don't know. And losing the virulence, we have the VBNC, variable state, but VBAC, non-culturable. Yeah. It's viable. The cells are here, but it's not culturable. And it happens that sometimes you receive plans to diagnostic for the government, and you try to isolate the bacteria. There's nothing. And you run a PCR on the plant extract, and you have a result, and you see phenotype 1, like, oh, what I'm going to tell to the government. Is it DNA? Or is it a living cell of Ralstonia that is in the sample? I don't know, but it's not culturable anyway. It could be. They lost their virulence. There was the ability of being cultured. They adapt so fast. It is, but finding the key for avirulence would be great for farmers and to prevent the bacteria spreading and the disease. That would be awesome. Yeah, we need more money, please.
Pregunta.
Voilà.
Soy como Mijael.
Pregunta. Maybe more than a question, I just remember that the topic related to the virulence of the presence of a viral and the strains of Ralstonia. I remember that 2017 generale from
Yes, CCG.
But the thing is that they decide that there is a relationship between the growing conditions of Ralstonia with the factor that in that kind of growing conditions, there are susceptibility to make expressions of the genome, the phenotype conversion system of Ralstonia, which is the main virulence factor of the bacteria and the cellular monocyte. So once in that tradition, this is the strains are vulnerable to this kind of phenotypic changes, and for that reason, the irreversible lack of the loss of the virulence strain. Yes, I have a question. It is more related to the divergences of the strains, because during my thesis, the most that I find is the secular relationship. During the thesis, we were working on MLPA, but I'm curious about that divergence you showed in one of the slices. When you're looking at the secular, you're looking at the secular diversity and also
MLST and MLVA, yeah.
Yeah, so it is most like a simple question because I was really curious about the big amount of divergency you can find inside of the secular. And how do you make it, how can I say it, comparable with other things in the literature? Because I have already noticed that they have specific codes.
Yeah. Those are MT or ST, but they do not have any capacity or I don't know how to say that is representing a systematic definition. Systematic definitions like species, subspecies, and then that is, I don't know. It's different. They're clones. It could be empty. It could be something else, but we're not ready yet to see that. For instance, the Escherichia coli, they are still using antibodies. O157H7, which is a different clones from the other one. He's causing check in the belly. So those characterization are working definitions of what a clone is and the relationship with the others. But it looks like MT, you have a huge amount of different MTs for one sequevar, but it's not systematic. It's typing and systematic. And they are complementary to understand what you're working with, but it's not the same definition. Okay, this is very different. So we're not using the MT actually to talk about a specific strain. People already have a hard time to work with sequevar. So imagine the MT, like 350 different MTs. No, we're not ready, and only for the phenotype 1. not for phenotype 2 or 3 or 4. This is very different. But we can use them for understanding, for instance, the 2B1 strains that we find in Chile. Are they coming from Chile? Are they coming from the Andes? So the birth of this specific lineage, or are they coming from Europe? If they're coming from Europe, that means that there are introductions of strains. Then you tell the government, okay, there are introduction of strains, we have to do something. Okay, this is very important. Or you, Chilean people, are you sending us strains in Europe? Okay, could be.
Thanks.
Tenemos una pregunta de Marcelo González. She said, thanks, this is my question. What are the prospects for the application of transcriptomics in the detection of Ralstonia solanacearum? Are these techniques reliable, especially in the field of international commerce regulation?
Well, transcriptomics, we use these techniques so to assess if the proteomics, the proteome of a given strain is the same as the other one, and to resolve some problems within the diversity of the strains. Proteomics has been used also to assess for a given group of bacteria the genes which were responsible for the host specificity of a given group. For instance, we have a group of bacteria that is wilting tomato and banana. And they have, phylogenetically speaking, another group of strain very close that can wilt tomato and cucurbits, but not banana. So we have a common strain, a common host, which is tomato, and differentiates hosts like banana on one side and cucurbits on the other side. Using this approach of assessing the nature of the activation of the genes, we could differentiate them pretty much and have the genes or the clusters of the genes responsible for this host specificity on banana or on cucurbits, meaning that using genome minings, we were assessing the two different groups and we found like only 9 genes different. The others, like 28 of the genes, were pseudogenes, so only 9 different genes, but they were not involved in this pathogenicity stuff. So this approach can use to resolve this kind of question. But the proteomics, yeah, it's fastidious also. Working with RNA and things like this, and sequencing RNA, this is a very delicate a molecule. Yeah, this was the term. Very delicate molecule, and yeah, it's a lot of work, and you have to be close to a research center that is sequencing it. But it was pretty cool to assess for us the host interaction with the bacteria and the genes responsible for the host specificity.
Bueno, no tenemos más preguntas. Agradecemos a todas las personas que asistieron, tanto presencial como que se conectaron en el Zoom. Hay preguntas muy interesantes, muchas gracias por las preguntas. Y entonces agradecemos al doctor Gilles Cellier por su presentación.
Gracias.